Okay guys, these are going to be some quick sketch notes to get you through most of Unit 6 that I copied over from the course exam description for AP Biology. So I'm going to read through some of these. I'm going to make some notes on some of the diagrams just to point out some integral parts and some important information that I think will help you understand some of this material maybe a little bit better. After looking at your guys' ed puzzles and your Khan Academy participation, I really appreciate you guys taking a look at those sources and participating with the questions that were on there. It looks like you guys have a good grasp of the basics of DNA structure and a little bit about replication, but I just wanted to kind of lay the information framework or foundation a little bit better just so you have something else to look at and something else to listen to. All right, so some of these that are on this list are the key aspects that you're going to have to know. These are the integral things that you're going to need to be able to apply knowledge of on the exam itself in terms of Unit 6. And these are just DNA replication, transcription, and then translation. So we're going to walk through all of those parts up until 6.4. So the first part of this talks about DNA, RNA being the primary source of heritable information. So you have to know that genetics can be contained in one of these two sources. So for most organisms, it's DNA. So prokaryotes, multicellular organisms like ourselves, and then in some cases, it could be in the form of RNA, which RNA we're talking about quite a bit because this is the genetic component of viruses. Viruses pass their genetic information on in a coded RNA sequence. Prokaryotes, in comparison to our double helix DNA, which is dual-stranded, prokaryotes have a circular chromosome. Okay, and we have that multilinear chromosome, meaning that we have the two strands. There are multiple locations for replication to occur. It's a much bigger molecule to have to deal with. That's why the process takes much longer than prokaryotes. Prokaryotes can go through the cell cycle in mere seconds compared to our process. So we take a look at the picture down here. We see a bacterial cell. This is their circular DNA. Okay, and there's also some genetic information, and we talk about bacterial transformation using this little source of DNA or genetic information to our advantage. These are called plasmids. Plasmids are circular DNA molecules that we have found utilization in genetic engineering. So we'll talk a little bit more about these later. You just need to know as of right now that bacteria has a circular DNA as well as these smaller circular pieces called plasmids. Okay, and these are used in genetic engineering. When we talk about GMOs, we're going to see how important this specific molecule here that's used from DNA is to making genetically modified organisms. Okay, so that's the first part. Let's talk really briefly about structural things, and these are just going to echo what you've already learned from the specific videos on Edpuzzle. But I found this little mnemonic here that talks about the difference between purines and pyrimidines. So we know base pair rules. You know that A pairs with T, C pairs with G. Some of your guys' word phrases for remembering those two pairing combos were pretty funny. Some of you with literal things, some of you were pairing like animals and dinosaurs, which I thought were really cute. But whatever, allows your brain to remember how to pair those two together, it's fine by me. One thing that you do need to differentiate between the two are structural types called purines and pyrimidines, and these are just bigger structures versus the smaller structures in the pairings. So I like this mnemonic because you guys can remember this specific elemental reminder about gold. Gold, the symbol for gold is Ag on the periodic table. Okay, if you can remember that purines are as pure as two gold rings, maybe that will help you remember that A and G are a two-ring structure. Now, I didn't think of one as witty or interesting for pyrimidines, but if you can remember that A and G are purines, okay, pure as two gold rings, that might help you to classify those, and then the remainder of them are going to be your pyrimidines. So your pyrimidines are your single ring structures. There's cytosine, there's thymine, and in RNA only there's uracil. And we'll talk about where this molecule comes into play specifically when we're making RNA from DNA. Okay, so those are just some basics about DNA structure. Okay, on to DNA replication. okay? So in years past, there were a lot more components that were going to be integral to you knowing the specifics about DNA replication, but since the redesign and even since the release of the core exam description, they've cut quite a bit of the fat, per se, out of DNA replication. Okay, so we're gonna talk about just some of the specifics that you're gonna need to know. Okay, so DNA replication. Obviously, we need this in order to pass on hereditary information. This is for mitosis and meiosis. We know both of those processes have DNA replication in interphase, specifically the S phase. That should be burned into your brains right about now. When we're making DNA, we're synthesizing in a 5-to-3 direction. Now remember, they were talking about specifically the deoxyribose on the DNA structure of the nucleotide. You can count these carbons, and the direction that the carbons are counted, that's specifically what they're talking about. If it's the first carbon that you're starting with, or the second, or the third, or the fourth, or the fifth, it's talking about the position of the specific molecules attached to those carbons on that sugar, okay? But what you need to know, specifically for these two numbers here, we're synthesizing, we're building the new DNA molecule in this direction, from the 5' end to the 3' end, in direction or in response to the new strand. Okay, and I'm going to talk about that a little bit more on the picture that we're going to label and discuss a little bit, but it's in respect to the new strand. It's the strand we're building, not the parental strand, the strand that we were reading. Okay, so replication itself is going to be called a semi-conservative process. What that means is it's half old or parent strand and half new. Okay, how are we able to make that new strand? It's because of those complementary base pairing rules. It's because we know that adenine and thymine go together. It's because we know that cytosine and guanine go together. Okay, and that's something that was established or coined by the two scientists Watson and Crick. Okay, these two individuals compiled with information from Rosalind Franklin, you know, her x-ray showing the DNA double helix. That's what gave us those two specific strands being formulated into its model, showing base pairs, showing the sugar and phosphate background, and showing that they go in opposite directions. That's what was specific to their model. All right, so some things that you're going to need to know or be able to recognize or specifically know their function in terms of DNA replication are the enzymes helicase, topoisomerase, DNA polymerase, and ligase. Okay, these four are going to be integral to your knowledge of the process and being able to recognize what's happening if you were to look at a simulation of it. Okay, so let's transition over to a diagram here, one of my favorite diagrams because it has all the working parts and it's very colorful and pretty to look at. Okay, so we're going to start from the beginning. Now, if we are looking at this specific molecule here, okay, we're going to begin with helicase. So helicase is what's going to unzip the parental strand. Okay, it's allowing access to both of those strands there from the parental strand in order to build our two identical molecules. Okay, what topoisomerase is, it's opening up or it's keeping that DNA helix straightened. It's unwinding it or it's unspiraling it. Okay, so this is going to keep the molecule from spiraling. So that's the main job of helicase, open up that helix, okay? In your specific Edpuzzle video you watched, you learned about SSB or single-stranded binding proteins. These are gonna hold the DNA open. So these other enzymes are able to access the molecule and to build that complementary strand without having to worry about the helix retwisting or reattaching, because it's held together by those hydrogen bonds. And even though they are weak bonds, there's still that attraction there between the electronegative oxygen and the hydrogen on the complementary base pairs. Okay, so one that you're not going to have to specifically know a ton about, but you should recognize, are these pieces here, DNA primase and RNA primer. So just as you would prime a wall in your house before you paint it, you know, ensuring that you're going to have a more even, smooth finishing product, DNA primase is going to make the job of DNA polymerase a little bit easier